Thank you, Rachel, and thank you everyone for coming. I want to say it was such a nice invitation to join this event here, and also to the CSW staff for organizing it so well. It was completely flawless. I'm here. The plane, there was some turbulence, but it's not your fault. Okay, so Banu and I are going to be presenting, and our talks are kind of related, so we kind of wanted to frame our discussion. So my talk and Banu's that follows, they're in conversation with each other, in part grounded in previous work that we have done together as biologists engaging with the newly emerging field of feminist new materialisms. We're happy to discuss our critiques and approaches to this in the Q&A, but broadly, both of our papers are attempting to re-engage with matter not as the uninterrogated products of the biosciences, but as biologies and matter grounded in their geopolitical context. So I just wanted to put that framing out there, and I will begin. This coming December marks the 30th anniversary of the gas leak tragedy that occurred at the Union Carbide Corporation's pesticide plant in Bhopal, India. Despite the Indian government's attempts to suppress information, media reports and scientific studies continue to draw attention to ongoing biological tragedies occurring in the city. These biological tragedies are widespread, affecting plants, animals, humans, and the local environment. In the surviving human population, there is all too much evidence of the world's worst industrial chemical disaster in the form of abnormally high cancer rates, respiratory problems, and excessive reproductive harm occurring at many different levels. The population of Bhopal continues to experience alarmingly high rates of stillbirths, miscarriages, and incidences of children born with genetic dispositions that are deemed as disorders or abnormalities by the medical community. Due to the initial exposure to methyl isocyanate gas during the explosion, and due to the further exposure of residual chemical contaminants in the soil and groundwater left after the disaster, the status of reproductive health continues to be an issue for both survivors of the Bhopal tragedy as well as new generations of local residents. Fears of long-term genetic and reproductive damage to the population have emerged, and many survivors have called for continued scientific research, reproductive monitoring, and genetic screening of the victims and their families. It is clear that methyl isocyanate, or MIC, and its metabolites have left their mark and continue to interact with the hormones, tissues, DNA, both somatic and gametic, and reproductive productivity of the Bhopali population. As methyl isocyanate continues to exert its effects on the reproductive biology of women, men, and children in Bhopal, remarkably, there is also a growing market of in vivo human labor developing in Bhopal that depends precisely on commissary futures made possible by the very same potential for reproductive productivity. Embraced by the convergence of neoliberal and capitalist ideals, reproductive tourism and surrogacy services have spread at an amazing rate across India. It is, however, particularly baffling that while the survivors of the Bhopal gas tragedy are denied proper aid and attention to their reproductive health concerns, biotechnologies of reproduction, including in vitro fertilization, gamete intrafallopian transfer, intracytoplasmic sperm injection, pre-implantation genetic screening, and gestational surrogacy are growing steadily in Bhopal. These biological and genetic microprocesses of life and labor are unfolding not apart from, but in direct relation to transnational circuits of capital supported by the postcolonial, neoliberal Indian state, the same state that opened its doors to foreign investment several decades ago, including multinationals such as the Union Carbide Corporation. Even though methyl isocyanate and its metabolites continue to poison a population, and most women in the region are being denied basic health care, Bhopal has managed to become a fertile ground for reproductive tourism, and a place where new reproductive and genetic technologies are able to thrive. In this paper, I attempt to conduct an analysis of entanglements and ruptures, genetic, reproductive, and otherwise, and contribute to an interdisciplinary conversation between feminist science studies, postcolonial STS, and new materialism, emerging primarily from feminist engagements with post-structuralism and cultural theory. I'm interested in developing a contextualized and situated materialist account of the chemical, physical, and political reactions of methyl isocyanate that also takes into consideration the active push and pull, and this is a vague way of not quite saying will, agency, and intent, exerted by this chemical compound onto human lives and its other surroundings. Methyl isocyanate gas was produced in the Union Carbide plant in a highly exothermic reaction, but in turn, to this day, continues to serve as a catalyst for further biological, biochemical, and biopolitical reactions. Prompted also by Michelle Murphy's idea of distributed reproduction and the chemical infrastructures of reproduction, I want to think about the role of methyl isocyanate in assisting or altering reproduction. Murphy suggests distributed reproduction as a way of reframing what counts as reproduction. She states, why should reproduction end at our bodies? How to participate in and challenge the ontological politics of reproduction. What is the place of industrial chemicals in reproduction? What is the place of industrial chemicals in reproduction if we now live in an era which some scientists have named the Anthropocene, a historical period when all life, all ecosystems, and the entire planet has been rearranged by human activity? Although my approach and reasons for studying the action of MIC are aligned more closely to discussions in critical posthumanism rather than that of the Anthropocene, I'm interested in opening up what gets to count as reproduction and examine the biological and genetic traces that are being left behind by this compound, both in the maternal and fetal environments and beyond. Related to this inquiry, I want to think about the chemical MIC in the context of the biological labor of surrogate women in Bhopal. Keeping in mind that infrastructures, including chemical ones, also aid in social sedimentation such as colonial legacies, the repetition of gendered norms in material culture, or the persistence of racialization, I intend to track the processes and outcomes of movement by hormones and proteins into the fetal environment from the surrogate, as well as the movement of fetal DNA into maternal surrogate blood. By tracing the breaking down of Bhopali survivors' reproductive bodies, their genetics, and their liquids due to MIC exposure, and simultaneously examining the building up of new fetal bodies through the genetics and liquids provided by Bhopali surrogates, my aim is to explore the compound MIC as a catalyst for the transplacental migration of transnational circuits of power and biopolitics. Context matters. Before tracing the active material life of MIC, I want to turn to the theoretical tools offered by new materialism, feminist science studies, and postcolonial SDS that will ground my analysis, and review a pertinent conversation ongoing between these fields. All three fields have turned to questions of materiality, and this material turn, as conceived in relation to post-structuralism and cultural theory, stems from a very specific conversation within feminist theory, which privileged postmodern constructivism and the significance of language to such an extent that matter appeared to be discursively constituted. Judith Butler's work is often taken as emblematic of feminist theory's flight from nature, or indeed its failed materiality. New materialist scholar Vicky Kirby has also raised a number of critical challenges through her close readings of Butler's analyses of nature, culture, discursive ontology, and conceptions of materiality. Turning Butler's proposition that sex is always already gender on its head, Kirby asks the risky question, what if culture was really nature all along? Indeed, Kirby explores the possibility that signs are substantively or ontologically material. Drawing examples from medical research and genetics, the cognitive sciences, and immunology, Kirby suggests that life itself is creative encryption. Calling attention to the agency and intelligence of biological codes, Kirby suggests that, for example, that the code cracking and encryption, this is a quote of hers, code cracking and encryption capacities of bacteria as they decipher the chemistry of antibiotic data and reinvent themselves accordingly might well be considered language skills. In a similar vein, I'm interested in calling attention to the skills of the compound MIC. In order to develop this analysis, however, I wish to both align and contrast Kirby's approach to calling forward the capacities of bacteria to my own attempts to animate our understandings of methyl isocyanate. In suggesting that bacteria have language skills, Kirby seeks to persuade a particular audience of feminist theorists committed to a transcendental humanistic frame that language is biological and biology has communicative capabilities. To support this post-humanist effort to acknowledge the communicative capacities of non-humans and to consider life itself as a text, Kirby compares two contrasting interpretations of Derrida's claim that there is, and quote, there is no outside of text, end of quote. The first interpretation reflects the view of many critics of post-structuralism who assert that, and Kirby states, we are caught in an endless slide of a referral that leads from one signifier to another signifier, one meaning to yet another meaning, in a spiral of implication that never quite arrives at its destination. As a consequence, we can never retreat or advance to some natural, prediscursive, or extra-textual space. As an alternative interpretation, Kirby appeals to the worlding of the world as, and I quote, a writing in the general sense, which articulates a differential of space-time, an inseparability between representation and substance that rewrites causality. It is as if the very tissue of substance, the ground of being, is this mutable intertext, a writing that both circumscribes and exceeds the conventional divisions of nature and culture. If we translate this into what is normally regarded as the matter of the body, then following Derrida, the most elementary processes within the living cell are also a writing, and one whose system is never closed, end of quote. Kirby makes a compelling case that cells write, and that it is in the nature of nature to write, to read, and to model. The ontological openness of the stance accommodates feminist theories of agency and subjectivity that have made their way into feminist science studies through, for example, Haraway's conception of nature cultures and Rosie Berardi's account of posthumanism and nomadic subjectivities. I myself am compelled by Kirby's suggestion of a more open ontological stance, and am interested in thinking about the compound methyl isocyanate as a chemical agent with agency and abilities such as writing, catalyzing, and imprinting. Nonetheless, Kirby's exciting ontological reorientations through feminist new materialism seem to omit critical ontological and ethical conversations that are ongoing in feminist science studies and critical modes of analysis in postcolonial STS, including, and these are some points that in a paper that Banu and I have co-written and it's forthcoming, we draw from the work of Suman Seth and other postcolonial STS scholars to develop these 4 points, or 5 points. The first is the recognition of situtatedness and the development of molecular materialist accounts of the world that are cognizant of matter across scales. The second is the articulation of local points of interest within larger networks and transnational circuits of power. Third is the detection of contact zones of empire. And 4, the destabilization of the central to become another local. These omissions raise important questions about whose interests are served by this new ontological terrain. To gain insights into the import of this omission, I believe it is helpful to return one more time to Derrida. Many scholars have suggested Derrida's original phrase, il n'y a pas de hors texte, is better translated as there is no outside text, rather than there is no outside of text, or there is nothing outside of the text. Derrida later noted in Limited Ink that the phrase, and this is a quote from Limited Ink, the phrase, which for some has become a sort of slogan, in general so badly understood of deconstruction, there is nothing outside the text, il n'y a pas de hors texte, means nothing else. There is nothing outside context, or that nothing exists outside context. It is the lack of context in Kirby's interpretation of Derrida in relation to writing and nature that is troubling. As critical as Kirby's ontological intervention is in extending language, reading, and writing skills to the non-human, it rings hollow without context. At its best, feminist science studies scholarship is immersed in the practices and data of specific sciences. The richness and credibility of science studies in STS depend on systematic knowledge of minute details of particular fields. Yet this specificity is not apparent in Kirby's references to the code cracking, encryption capacities, and cryptographic skills of bacteria. What is lost in the absence of this specificity? New materialism takes on quite different meanings in the context of feminist science studies and postcolonial STS than it has in its encounters with postmodernist philosophy and cultural theory. A long tradition in feminist science studies has been driven by social justice epistemologies, which recognize that certain bodies, such as people of color, reproductive bodies, disabled bodies, animals, plants, and others subjected to colonialism, racism, capitalism, patriarchy, and science, have been inextricably tied to nature. Not all feminist and postcolonial scholars have had the opportunity or the desire to join the flight from nature or from their biologies. A feminist STS and postcolonial STS approach to discussing the ontological status of bacteria as writers, readers, and modelers, or MIC as an agent that writes, catalyzes, and imprints, would be, for example, to discuss these biological and chemical agents in a context that is structured by the domains of the life sciences and capitalism, or by what Karthik Sundararajan has referred to as biocapital. Similar to their human, animal, and plant counterparts, in the purview of the life sciences such as genetics, recombinant DNA technologies, and in particular synthetic biology, bacteria, for example, not only carry information, but they are information. The sign becomes ontologically material here, but only through its labor. However attractive it might be to imagine bacteria's writing and language skills within a very different ontological orientation, as life sciences are practiced under contemporary capitalism, bacteria's skills conveniently serve a highly mechanistic view of life, which not just figuratively, but literally forces bacteria to write. In the realm of synthetic biology, for example, bacteria such as E. coli are being purposely bred to write. This writing consists in transcribing DNA and translating RNA, purposes that serve human interests alone. Once they perform these tasks, the bacteria are promptly snuffed out for their valuable proteins. This is not the high-profile life of cryptographers and code breakers. More akin to the value of natives in the jungle for colonial science, bacterial writing becomes bacterial labor that is harnessed and controlled, and its products managed and turned into profitable use through the imposition of order and predictability. And that last line is a quote from Kavita Phillip. So in the context of bacterial writing, Melinda Cooper's work in Life as Surplus poses for us another question. She asks, where does reproduction end and technical invention begin when life is put to work at the microbiological or cellular level? Discussing biological growth in the context of neoliberal biopolitics, Cooper suggests that neoliberalism reworks the value of life by effacing the boundaries between the spheres of production and reproduction, labor and life, the market and living tissues. In the context of bacteria, then, it is impossible to isolate and appreciate their special talents, including writing, outside the spheres of reproduction, production, and labor. Is there a way, then, to think about the work done by methyl isocyanate as ontologically material, but also avoid the problem of decontextualization? Is there a way to ask how, as a chemical agent produced and activated by neoliberal practices, MIC keeps the flow of biocapital alive and reworks the value of life by effacing the boundaries between the spheres of production and reproduction, labor and life, and the market living tissues? By incorporating social justice epistemologies informed by histories of colonialism, class politics, civil rights movements, women's reproductive and health movements, I want to end now by returning to methyl isocyanate and gesture towards important ontological understandings that can be sustained through practices of contextualization. Broken lines, broken lives. The Bhopal gas tragedy occurred in the early morning hours at the Union Union Carbide Corporation's pesticide plant in the city of Bhopal. One of the chemicals used to produce Union Carbide's pesticide 7 was the compound methyl isocyanate. There is, of course, controversy around the cause of the accident, but what is known is that workers at the plant that night were cleaning pipelines around a methyl isocyanate holding tank. As they had likely done countless times before, water was used to wash away the grime from the pipes. On that particular occasion, however, water leaked into the holding tank, where the methyl isocyanate was housed. When the water met the chemicals, the following chemical reaction occurred. This highly exothermic reaction resulted in the emission of 27 tons of methyl isocyanate gas into the night air in a matter of hours. According to the government of Madhya Pradesh's official Bhopal Gas Tragedy Relief and Rehabilitation Department website, vapors of the methyl isocyanate gas lofted throughout the night air in a southeasterly direction, reportedly killing about 3,000 people almost immediately from the toxic fumes. Independent and non-governmental agencies claim, however, that this number is grossly underestimated and that closer to 8,000 to 10,000 people died during the first weeks following the exposure to gas. Survivors remember their bodies being turned inside out on the night of the disaster. Those who died immediately were bringing up bodily fluids and were reported to have been foaming at the mouth. This next slide is a little disturbing. Just wanted to warn you. The chemical also acted as a severe irritant of the eye, so that tears flowed constantly. It is reported that thousands of animals died in the immediate surrounding areas of the chemical plant, and that leaves on the surrounding trees of the Union Carbide plant turned yellow and fell to the ground. In the years that have followed, at least 100,000 to 200,000 more have suffered from permanent health damage. As of 2014, members of the International Campaign for Justice in Bhopal claim that the Union Carbide disaster has killed more than 25,000 people. This estimate includes not only the immediate deaths resulting from the gas leak, but also deaths caused by the effects of ongoing water contamination resulting from the inadequate environmental cleanup efforts in the aftermath of the gas leak. Drinking water is still contaminated in some parts of the city that had been directly affected by the gas in 1984. 30 years later, some have no choice but to still drink from the groundwater in these areas. MIC gas and reproductive health. The majority of those who were affected by the initial exposure to toxic fumes were the poorest of the poor living in slums around the carbide plant. Those who were able to escape the deadly fumes by car were later noted to have had better survival rates and experienced fewer long-term health problems. But those without vehicles had attempted to run away from the fumes on foot, inhaling the toxic air. In the wake of the explosion, the toxic methyl isocyanate gas, which was heavier than the surrounding air, settled low to the ground, thereby exposing smaller children to even higher concentrations of fumes than were the adults. Methyl isocyanate, known to be a highly irritant chemical, is absorbed through the skin and reacts with various liquids and mucous membranes in the body. It also has the ability to penetrate through tissues and react with proteins in the body. In 2000, the International Medical Commission on Bhopal revealed a list of conditions which they deemed as the most pertinent long-term health issues faced by survivors. These conditions include ocular problems, respiratory toxicity, reproductive toxicity, genotoxicity, and carcinogenicity. Immunotoxicity, it doesn't end, sorry. Psychological and neurobehavioral toxicity, and neuromuscular toxicity. Though the long-term health conditions experienced by the survivors are many, I want to leave you with a focus on the reproductive health in women and genotoxicity observed mostly in the women and children in Bhopal. Women who were in their reproductive years during the time of the gas disaster immediately started to experience the effects of methyl isocyanate poisoning on their bodies. Women who were pregnant at the time of the gas leak experienced spontaneous abortion rates that were 10 times higher than the normal population. In 2009, a review of clinical and experimental findings from the Bhopal tragedy reported that menstrual, and I quote, menstrual abnormalities, vaginal discharge, and premature menopause have emerged as common problems among Bhopal MIC-exposed women and their female offspring or girl children. Not only has the chemical affected the immediate survivor population, but in line with chemical infrastructures that Murphy talks about that assist or alter reproduction in a distributed sense, MIC is transforming life in intergenerational time. Enough time has now passed to observe long-term genetic effects of methyl isocyanate exposure. Of the very few studies that can be accessed, several cite the presence of chromosomal aberrations. These aberrations appear in the form of breaks and gaps in the DNA of cells such as lymphocytes. The incidence of spontaneous abortions may also be related to the issues of genotoxicity. The direct effects of methyl isocyanate on gamete development and zygote formation have not been studied in sufficient detail. Of the studies that do appear on the effects of MIC on gametes, only male germ cell and sperm mutagenicity are examined. In an in vitro study using a male mouse spermatogonial cell line, it showed that MIC causes DNA damage, oxidative stress, and apoptosis. Chromosomal aberrations were also reported in the form of telomere anomaly, aneuploidy, and variable amplification of microsatellite repeats. One study that has examined a mouse ovarian epithelial cell line reported that MIC exposure resulted in ovarian carcinogenesis through persistent DNA damage. This damage included telomeric dysfunction due to premature senescence and plausible association with chromosomal and microsatellite instability. Interestingly, a recent retrospective review of cytogenetic studies on MIC raises the concern of epigenetic effects of MIC gas and the possibility of vertical transmission of genetic aberrations. MIC-mediated distributed reproduction. In their article, Twice Poisoned Bhopal, Chandana Mathur and Ward Morehouse explained the various elements that were necessary for the perfect storm of bodies, environments, and technologies that resulted in the chemical disaster in Bhopal. They and others have developed a link between methyl isocyanate gas, the explosion of the gas, and the Green Revolution, which took place in India during the 60s and 70s. This government-sanctioned initiative of the Green Revolution introduced the need for high-yielding seeds, chemical fertilizers, and therefore also the need for pesticides. The demand for the production of pesticides, coupled with the perceived expendability of the poor residents of Bhopal, provided the perfect conditions for Union Carbide to build its manufacturing plant in Bhopal. As Mathur and Morehouse suggest, the Bhopali people faced the head-on impact of world historical and political economic processes. Transnational economic practices responsible for the Green Revolution in India and the presence of the Union Carbide plant in Bhopal resonate all too perfectly with the recent boom in reproductive tourism and surrogacy in India. The very same elements involving the ability to find cheap labor and the unwillingness of the Indian nation state to discourage foreign private investment appear to be at the heart of India's recent boom in reproductive tourism and surrogacy. Banu will talk more now on surrogacy in India more broadly, but I want to leave you with the thought that MIC-mediated distributed reproduction began long before the Union Carbide plant explosion and before the Bhopal gas tragedy unfolded. Therefore, to really appreciate the writing, catalyzing, and imprinting skills of MIC, it will require that we also recognize the transpositions and transformations of life, death, and matter in the context of MIC-mediated differential space and intergenerational time. Thank you.